So Brian comes to us, he has a PhD from the University of California at Berkeley and uses his expertise in disinfection and pathogens to address a wide variety of issues in water, wastewater, and recycled water. He's currently engaged in a number of projects related to potable and non-potable reuse, including the newly emerging paradigms of surface water augmentation and direct potable reuse. In this area, he works with the development of public health criteria for potable reuse, the design, evaluation, and testing of reuse technologies. And through these projects, Dr. Pecson has been working with the California Department of Drinking Water to advance and expand options for the design, implementation, and permitting of innovative potable reuse systems. With that, Dr. Brian Pecson.
So I'm going to be talking about pathogens today, which seems like a great topic given the person who has just won this award. So congratulations, Chuck. I started working 7 years ago at Trussell Technologies and was always in awe of Rhodes Trussell, just his breadth of knowledge on things. And so it struck me very strongly in the beginning when he spoke of you in those same ways that I speak of him. So on his behalf, Trussell Tech, the whole industry, thank you for all that you've done. Okay. Need to figure out how to use that. Okay, great. So, what has Chuck done? Well, he's told you some. The lecture last night was really a fascinating history as well of what Chuck has done, the role that he has played in bringing quantitative methods for us to look at this question of pathogen risk. So we had a history of how this was created back in the 80s and then integrated into our drinking water treatment regulations. This approach clearly has taken hold, and it's clear that we need to be using similar approaches to look at the safety of the next set of questions, which is potable reuse. So one of the key topics for today is direct potable reuse. And perhaps the most important questions facing us are whether or not we can be controlling pathogens. So let's start by looking at what we have done the most experience with, which is indirect potable reuse. We know how to do this safely, actually, and I can say that because we've been doing it for decades here in California. Because pathogens are so important, we've set very stringent pathogen control requirements. So on this slide, you can see that we are using the same pathogens in potable reuse that we use in drinking water as well, virus, giardia, and crypto, and those are the right numbers. I guess this is mostly a California crowd, so you've seen these numbers, 12, 10, and 10 logs of inactivation, where 99% reduction is 2 logs, so we truly are talking about 99 point, and then put 10 nines after that for virus. So how do we actually achieve these really high levels of inactivation? Most of our projects are putting a lot of emphasis on pathogen control at the advanced water treatment facility so that, let's see, it's a tough angle for me here, but so this facility over here, most of the pathogen control is happening there. And we do get some credit as well for the aquifer. But what we're finding out more and more as we study aquifers and the protections that they provide is that there's a whole lot more than we might have even thought happening. A lot of different mechanisms are going on to help control pathogens and their eventual passage to the people drinking this water. And so there's a number of studies going out. We've looked at some soil column studies. Here's a picture of the soil columns in our lab and started to try to quantify the amount of pathogen reduction that's actually happening here. So in the past, this has been more of a black box, what actually does the aquifer do, and we're starting to quantify this now. So you can see in these tables, just what's important here, we've been looking at a number of pathogens or indicators, in this case MS2 virus, and when we did 3 rounds of testing through these columns, you can see that we get really high levels of virus kill, so greater than 5 to 7 logs, and in the background as well with Cryptosporidium, in excess of 7 logs. And so what do we get credit for in the existing regulations? We get some credit for die-off of virus as it goes through the ground. We get a log of credit for every month that it's in the ground, but it's clear that there's a lot of other mechanisms going on here that are uncredited. And this is important because we see that this buffer, this aquifer, is really important at helping us to balance out any sort of treatment issues that we might have at the AWTF. It's going to get picked up by the environment. So this is in groundwater recharge and reservoir augmentation, the next form of reuse that's moving forward right now in California as well. There's a number of mechanisms as well that are protecting us if there's any sort of upsets here that are going to pick up the slack. One of the things that actually the only thing that we're getting credit for currently is for dilution and mixing. So here's some results from Padre Dam, who's doing a reservoir augmentation project in the San Diego County. And we can see from this modeling that even under worst case scenarios, water that goes into this reservoir is going to get diluted at least 100-fold, and typically much more than that. So we do have, again, this buffer in the environment that can help us even out some of the peaks. This is important because it gives us tolerance in the to accept some variability in the performance of the AWTF. Why? Because there's such a powerful buffer that's downstream of treatment that could help deal with any issues. So, is this going to be the same case in direct potable reuse? I would argue potentially not. So we have here the AWTF again. In the absence of environmental buffer, this water is going to go in. If there is any sort of upset in treatment, it's going to go unattenuated through the pipeline and to consumers, and this is important because these peaks could then make people get sick. And so when we think about the sort of reliability of treatment that needs to happen at this facility, it's different between IPR and DPR because we don't have anything intervening to help us pick up any slack. So this treatment has to be really effective 100% of the time. So if that's what the problem is, the question is, can we still do DPR safely? And luckily the answer, well, at least we have the green light to go ahead and keep exploring this. There was an expert panel that Chuck and Joan were a part of that looked at this question of whether it's feasible to do DPR in California, and the answer was yes. The expert panel recommended that we do additional research projects to understand some of the key outstanding issues a little bit further, and really what these research questions are related to is public health and controlling against the two main groups of contaminants, pathogens, and chemicals. And so I'm just showing now here how those pathogen research projects fit into the overall scheme of things. Really what we want to know is, can we make this water safe? And how do we determine if it's safe? What would we need to know? We would need to know how much pathogens are coming into our wastewater treatment plants, into these advanced facilities, and how well our treatment plants are doing at removing those pathogens. Because ultimately that will lead to some concentration in the water, and we'll have to determine, is that water then safe or not? And so you can see, just laying out what they have suggested, there's been an interest in looking at pathogen monitoring and outbreak monitoring, looking at pathogen concentrations in the sewage as it gets to the wastewater treatment plant, advanced water treatment facility. And then secondly, we want to see how well does this system perform, and can we reliably get this water looking good really all the time so that the people that are drinking this water don't get sick. And so the two research questions focus on these two things. How much is coming in? And how well are we doing at removing it? So let's look at the first question, pathogen monitoring and outbreak monitoring. Why is this important? Let's see. Yeah, I guess we haven't talked about that 12-10-10, but when we developed those criteria, the way that this was done was to look at the concentrations of pathogens that are coming in the wastewater, and then look at how much we want to have in the treated effluent in our drinking water. We'll define some level as acceptably low concentrations that we deem safe then, and we determine how much treatment we need by looking at the difference between what's coming in and what we want coming out. And so of course, if we have higher concentrations of pathogens in our wastewater, we're going to have higher degrees of treatment specified. And when we look at the existing regulations, or how the groundwater recharge regs were developed in California, for example, there is an assumption, because of lack of data, that these concentrations were very high. And so in the absence of knowing more about these pathogens, the assumption was this is really high, and so it leads to very high treatment requirements. Of course, this could be lower as well, and if we could demonstrate that they were lower, perhaps we would get by safely requiring less treatment. One of the problems is that we don't really know very well what these concentrations look like. So Joan did a great study in 2004 for Water Environment Research Foundation to look at this question of what are the loads of pathogens in raw wastewater, and then how are they removed through wastewater treatment plants. So Joan looked at a number of pathogens and indicators and developed this data set. This is really a seminal study for the industry, and it's still the study that's used today for help with permitting of facilities and understanding treatment requirements and pathogen reductions. But I think as Joan was saying in her talk just now, that you'd probably agree as well, that we need to be expanding beyond this. In this study, Joan looked at 6 water recycling facilities in Florida, Arizona, and California, and took 5 to 6 samples of pathogens and indicators. And what this graph here is showing on the right is the distribution of Giardia that she saw at these facilities. So on the top is the concentrations that she saw in the raw wastewater, and then on the bottom in the secondary effluent. But we need more information. Like this is a great data set and a great place to be starting, but we need more information beyond this to look at the impacts of things like location, seasonality, the size of your facility, the size of your sewer shed, and the type of treatment that you're providing as well. So the expert panel recommended that we further expand these data sets to know exactly what these levels are. And if we find that these levels are higher than we think, then we should maybe have higher treatment requirements. That's not what we're finding so far with the data. On the flip side, if these levels are lower, perhaps we have a rationale for saying, all right, let's consider reducing these requirements. Why is this important now? I mean, we've been doing potable reuse for decades. Why is this important now? And I think it ties back to the fact that with DPR, we really have no buffer in the environment, clearly. Literally, we have no buffer. So the degree of uncertainty, we want to be reducing that in each case, because we need to make sure now, as consistently as possible, that we're being protective of public health at this facility. So the industry is taking on this challenge. We're involved with a couple pathogen monitoring studies that are ongoing right now for the city of Oceanside and the city of San Diego. So I'm just overlaying some additional data that has been collected on top of Joan's data set here in orange. I think we have been looking at each of these locations, taking 16 to 24 samples in a variety of locations in the treatment plant, and then also looking at multiple pathogen groups. So the 3 classics that we've been talking about before, enterovirus, Giardia, cryptosporidium, but also other important pathogens that we want to be further characterizing as well, including norovirus, salmonella, and of course, a lot of the indicators as well. So the first point was that we need more data, but I think we need to stress as well, again, echoing what Joan was saying, that we need to also have really good data. When we think about doing these campaigns, there's going to be a big push. There's money from the state board to further investigate these pathogen concentrations, but we want to make sure before we invest in these expensive studies that our methods are actually as well adapted for this task as possible. And so what I'm showing here is a picture of the EPA method that's used for looking at crypto and Giardia, EPA method 1623. And this method was developed back when we wanted to be characterizing source waters for drinking water. So this is just a little clip from the introduction that says explicitly that this method was developed to look at crypto and Giardia in surface waters. Of course, now we want to be, so it was made for a certain matrix, and now the question is, are these methods that we already have suitable for testing of much different matrices like raw wastewater or secondary wastewater? So this is going to be a critical research need, so I guess it's great that there's so many university students here and industry. The development of these methods is going to be critical before we start embarking on these big campaigns. So just to tie back to that protozoa, crypto, and giardia method, there's a couple different ways that we can be taking these samples and processing them to get the solid material that we then look at under the microscope to quantify these pathogens. There's the existing drinking water method, 1623, that was developed for surface waters, and people have said, hey, maybe this isn't so great for a wastewater. And so they've modified the method with this EPA 1693. It says, let's get rid of this whole filtration thing. This water's so chunky anyway. Let's just centrifuge it and skip that step. We're probably losing a lot of pathogens in the way. And so these are a couple options for concentrating up pathogens from raw wastewaters or waters that have high concentrations of pathogens. Thank you. One of the things that we'd like to do as well is characterize how well we're removing these pathogens through a wastewater treatment plant, and those concentrations are going down as it goes through different unit processes, and we'd like to count how much that's happening. And so when we look in cleaner waters like secondary effluents or tertiary effluents, we need to be processing larger volumes of water as well. And so there's new methods that are coming out to look at these larger volumes and help us get detects in some of this cleaner water. And so this ultrafiltration method is another one of great interest. And so one of the things that we did for the pathogen monitoring study at Oceanside was, before doing all of the testing, which is expensive, was to do a comparison of these methods, comparing 1623, the blue, with 1693, the red. And this was done at Jones Lab in Michigan State. And what we found from doing this analysis was, despite the fact that this red 1693 was made for raw wastewaters, when we did the actual comparison, we saw much better recoveries in most cases for both crypto and giardia using the drinking water method. And so we're probably just scratching the surface on this whole question of pathogen detection in these waters, and I'm trying to emphasize that this is going to be a really critical step for us so that we ensure that we're getting good data at the end. And I think there's hope for us to actually achieve this, since there is going to be, I think, significant funding from the state to further develop these methods and then build up this data set. So coming back to here, what we've just talked about is this first part. Let's understand what's coming in. And then the second part is, how well are we doing at removing these pathogens through treatment? And why do we care about this? Because ultimately, we want to understand what is the risk that people are going to experience drinking this water, this direct potable reuse water, and to figure out, is this water safe, in quotes. One of the ways that we do this then, of course, is with the topic of a lot of Chuck's work, which is quantitative microbial risk assessment. So just to place us back in the context, what do we care about? This is our new scenario. This is what direct potable reuse may look like, and we want to prevent this situation from occurring. We don't want hiccups here that send out red arrows, bad water, to our consumers. So how do we do this? Well, there's people that are doing this every day, and these are drinking water treatment folks. They have a system that looks a lot like DPR. They treat water, there's no intervening barrier, and they send it to consumers who don't get sick. And so are there lessons that we can learn when we move to DPR from drinking water? I think the answer is yes. And what we see is that drinking water treatment facilities are frequently designed to provide more than the minimum degree of treatment to prevent this possibility of off-spec water ever being produced and distributed. And so I'm just showing this drinking water treatment plant, and what we find is that there's multiple barriers that we use at these facilities, and frequently these barriers provide redundant protection. The first time I looked at designing a drinking water treatment plant, they said we have a disinfection system and a redundant disinfection system. I said, great, so the redundant turns on after the first one has a failure, and they said, no, these two are always on, which was a surprise to me, but they can't have failures. And so they set up a system that is nearly bulletproof. And so this is a concept that we use in drinking water treatment. And the question is this something that we can bring to potable reuse as well? And so we could try to answer this question by looking at the safety of two different design possibilities. One, we have a train that's designed with enough barriers to just meet the pathogen requirements. And then we set up another one that has those same barriers plus one additional barrier. And we want to understand this question of safety. How much safety does this provide? And really our access to answering this question is through QMRA, which Chuck has talked about so much before. So what are we doing with QMRA? We're looking at the concentration of pathogens that are coming in. We're looking at how well our treatment removes those, because ultimately some small number is going to be present in the water that people are drinking from a DPR scenario. So that tells us how much people are exposed to. We can link this exposure then to risk with these dose response curves, and then characterize the risk and figure out how many people are going to end up sick. What is the fraction of people that's going to be sick? What is the rate of infection, for example? So this is a super powerful tool because it allows us to look at the impacts of a lot of different things. What if there's an outbreak? These concentrations in pathogens are going to increase. What is that going to do to the number of people getting sick? Is there seasonality in these pathogen concentrations? Is crypto more of a winter disease, whereas Giardia is constantly present throughout the year? Is there differences in the regionality of these pathogen loads? I think importantly, as an engineer, these are some of the biggest questions for us as well, is in this middle part. What is the impact of treatment failures on risk? I think that this tool is a really excellent one for us to be understanding this question of redundancy. Like clearly, redundancy is a pathway to make safe water, and one of the outstanding questions still is just how much do we need to make this water safe? Because clearly there's a cost to doing this. We don't want to be putting so much that it's excessively costly, but we also don't want to be threatening public health. And then also, we want to be evaluating different treatment trains. We know that one works, for example. How would we assess whether another one would work? This tool could help us answer these questions. And so let's go back to this example with the two treatment trains. We can use QMRA to look at the risk then from these two different options. In the top, I'm showing the treatment train, one that has no redundancy, and in the bottom, the one with redundancy. And this is just a probability distribution at the bottom, and on the y-axis, this is risk. And so we're just setting a goal of 10 to the minus 4, which it was great to hear last night from Chuck that the origin of this 10 to the minus 4, I was really happy to learn that. Sorry, Chuck, that I'm showing this. Apparently, I thought you'd be happy, but it's actually the opposite. But regardless, we have this risk target, and you can see in both cases, both of these trains, either with or without redundancy, can meet the risk target. But what we find is that even brief failures in the unit processes that make up this treatment train can have really huge impacts on the risk profile. And so you can see in this case of a treatment train designed with no redundancy, even 15-minute failures would cause this risk profile to exceed our limit. Whereas the redundancy, as you might imagine, we have a failure occur in some of these processes, albeit brief, and still we see that we still have a lot of protection from failure, where failure is really understood as crossing this threshold. So we recently finished a study from a Water, Environment, and Reuse Foundation work looking at a demonstration project for DPR in San Diego. And what we looked at was a treatment train composed of, we took tertiary effluent, and then we had pre-treatment with ozone and biological activated carbon, followed by the full advanced treatment train, MFRO, and UVAOP. And what we found was that this treatment train could provide a high level of public health protection, even in the face of treatment failures. And so this work help provide evidence that yes, we can do DPR safely, and a lot of the data from this project actually was provided to the expert panel who used it as well to determine that yes, we could move forward with DPR. It is possible. So we have a treatment train that works, which is this one. This isn't the only treatment train that works. There could be less treatment potentially and still provide safety in the system. And so these are some of the questions as well. The second kind of research thrust from the state that the state's interested in is using QMRA to understand the safety of different trains. And so that is something that will be maybe discussed at next year's meeting. So wrapping up, I think the future looks bright for direct potable reuse. We have a really serious threat, which are the pathogens, but both the expert panel and the regulators agree that we can continue moving forward with direct potable reuse in California. There is additional information that we could be collecting that'll give us greater confidence in our ability to do this, and that's what we've talked about today, looking at pathogen concentrations, performance, and risk to allow us to kind of quantify how well these systems perform. And once we've gathered this information, we can be using this knowledge to help develop DPR criteria that are protective of public health. And with that, I thank you, and we have time for questions.
You do have a couple minutes for questions, Brian. You do have minutes for questions? Okay, good. Anybody have any questions, Brian?
Great talk, I enjoyed it. So how do you balance the need for redundancy to increase, safety to the cost of the processes? Did you compare it, for example, when you had more units, let's say 4 units, to other alternatives like brackish water desalination or even seawater desalination?
Comparing potable reuse to some of the other.
All this redundancy, eventually the cost will be prohibitive, and I wanted to see how close it comes to the cost of other alternative to get water, like brackish water desalination or even seawater.
Yep. So I'll get to that. But maybe I'll first say that a comparison that's easier for me to make, for example, is to just think about DPR versus IPR. A lot of the cost for the whole system is associated with moving this water to different places. And so doing IPR with reservoir augmentation is going to have costs associated with pipelines to the reservoir, for example. And in the case of San Diego, which tested this treatment train and is now implementing it for surface water augmentation, they had the possibility of using a reservoir that was much closer to their wastewater treatment plant by having this additional treatment that's being provided. So it opened the door for new options, and these new options, when you look at the cost of the overall project, can be significantly less than doing less treatment with longer pipelines. And so I think that is one of the big carrots for DPR as well, is that the cost of these projects can be significantly less than some of our indirect potable reuse projects because of this reduction in infrastructure costs, which can sometimes be much larger than the cost of treatment. As far as comparison with desal, there's the much higher costs associated with the RO in these cases with higher TDS water, for example. So energy-wise, there's savings potentially doing potable reuse instead of desal. I think beyond the cost of those, the practicality of setting up these systems and getting permitted to have a new discharge, for example, into the ocean are going to be some other factors that need to be weighed. Just the implementability of these options. I think there's a lot of work going on that's looking at some of these analysis of different forms of reuse with and without RO compared to desal, et cetera. I think the WENRF has a lot of studies or a few studies that are looking at this topic, and so I think there's resources there too.
Time for one more, Andy.
Okay. Hey, Brian. So you pointed out that the failure of a process can drive the risk, and you pointed out that there's a large safety factor imparted by, for example, the groundwater recharge and the pathogen removal. 5, 6, 7 lot of logs on there. Can you comment on how necessary that safety factor is, and should that be really better incorporated into the overall log requirements? Going back and recognizing that we've had 30 years of successful implementation.
Yeah, I think definitely that we should be getting credit for everything that we feel good crediting. And probably the Aquifers been a black box more so till now. We don't get much credit for any processes going on in there except for viruses. And almost assuredly, probably regardless of conditions, there's pathogen reduction that's going on. For us to be able to quantify and credit that in a way that makes everybody feel comfortable should be one of our big goals. It's interesting as we move to direct potable reuse that, you groundwater recharge, so much is happening here that we're not crediting. That's really a great thing. The aquifer is such a great buffer. And it's clear with direct potable reuse that we don't have that. And so I think it will change the way that we design these facilities. But like you're saying, we should be getting credit for everything. I think these pathogen monitoring campaigns that people are doing to better understand wastewater treatment plants as well is a critical thing, because right now we're probably undercrediting a lot of wastewater treatment plants when if they're removing pathogens, then let's get credit for it.
Thanks, Brian.